Hello, everyone. Welcome to the Memorial Sloan Kettering Science Spotlight Series. My name is Omar Abdel Wahab, and I'm a faculty member in the Human Oncology and Pathogenesis program in the Department of Medicine at MSK. This seminar series features lectures from today's leaders in basic and translational biomedical science. If you have questions throughout the talk, you can email them to the Office of Scientific Education and Training at, David, could you hit the next slide? At oset@mskcc.org, as you see at the bottom of the slide there, or you can tweet your questions to @MSKEducation, which is also listed at the bottom of the slide displayed right now. For the best viewing experience, please use Google Chrome. If you're using Internet Explorer, please use version 11 or higher. If you have any technical issues during the live stream, please lower the video resolution, and you can do that by clicking on the gear shift on the lower right-hand side of the screen. Okay, so it's now my absolute pleasure to welcome our speaker for today, physician scientist Dr. David Solit. David is the director of the Marie-Josee and Henry Kravis Center for Molecular Oncology at MSK, as well as the chair of the Geoffrey Beene Cancer Research Center at MSK. He's also a member of the Human Oncology and Pathogenesis program. As many of you likely know, David's work has really been instrumental for so many initiatives at MSK linking molecular genetic abnormalities to targeted therapies for patients with cancer. And I should say that his work has not only affected patients at MSK, but also internationally. So really look forward to his talk, which is entitled today, Defining the Actionable Cancer Genome. Dave, thanks so much, and the floor is yours.
Thanks so much, Omar, and thanks to the organizers for the opportunity to present our work. I would start by saying that the work that I'm going to present today is really the efforts of a large team, probably more than hundreds of individuals, probably thousands of individuals at Sloan Kettering, clinicians, translational scientists, basic science researchers, who have really been unified into the goal of trying to use a better understanding of biology and genomics to improve the care of our patients. So I'll just start by acknowledging that I've been a consultant for several companies, including Molecular Oncology, Lilly Oncology, Pfizer, and others. So the focus of my research efforts, and I've now been at Sloan Kettering, this is going to be my 23rd year upcoming, and I've been running my own research group for almost 15 years now, has really been to try to accelerate drug discovery for patients with cancer. And we take what's called a precision medicine approach to this. What we try to do is we try to identify targets in patients. These are usually genetic mutations or other alterations that are important for the development of the tumor and the progression of the tumor. And I'll go through some time trying to explain how we identify these targets and how we leverage them for drug development. One of the major challenges is identifying a drug that inhibits a particular target, and that's not something that I spend most of my effort on. There, I would say about 2/3 of the researchers at the institution are trying to do this here. But what I really focus on mostly is trying to identify the patients who have an alteration in a particular target or set of targets so that we can, in a personalized and individualized way, match that patient up with the correct drug or combination of drugs that are going to be most effective for that individual's tumor. And so how do we define these targets? There's really two ways that we do this. One is called a genotype to phenotype approach, and this is a very statistically driven methodology. And what I mean by that is we usually, for these efforts, take large numbers of tumor samples or other types of samples, and we apply molecular or other profiling assays to these cohorts, and the assays that we use have obviously changed over the years and have become more sophisticated. But we really base the identification of these drug targets based upon their recurrence within the population. So something that's found multiple times in patients, different patients with, say, the same cancer, that recurrence is probably not coincidental. It's probably biologically, induced, and it suggests some sort of selection for that type of mutation. And it was really the identification of BRAF that really changed my career. I had been working on a target called HSP90, a protein that's very important in the biology of cells, and we weren't really making a whole lot of progress in trying to drug that. target when I read this paper in the summer of 2002, which identified BRAF as mutated in human cancer. And it was something that no one had really looked for in the past until this group here at the Sanger took a more shotgun approach to identifying potential new mutated targets. And so what they did is they took about 1,000 cancer samples. Most of these were actually cell line models, and they profiled those models and tumors for mutations in the RAS MAP kinase pathway. And we had known going into this work that RAS was commonly mutated in human cancers, but we weren't aware that almost 6 to 8% of tumors had mutations in BRAF, which was really the novel finding from this group. And what was notable was that there was a single mutation originally labeled V599E, later corrected to V600E, that was located within the kinase domain of BRAF that was found in about 95% of the tumors that had BRAF mutations, and in total in about 50% of patients with melanoma, less commonly in other cancers like colorectal cancer. So the fact that you would recurrently see the same exact mutation arising in different patients, especially in some tumors like melanoma at 50% incidence, really suggested that this was probably very important for the biology. So what happened was they went ahead and they made this mutation in the laboratory. They put it into non-transformed cells, and they were able to transform those cells into a more cancer-like phenotype. So the experiment that we did initially was we wanted to ask the question, and this will be very important to later in my talk, is that if you've got BRAF mutated in your tumor cell, is that cell now dependent upon BRAF? Because if the answer is yes, BRAF is a good drug target. Inhibiting BRAF might kill that cell or at least stop its growth. Whereas if the answer is no, the tumor is not dependent upon BRAF for growth or survival, targeting it is probably not going to have much effect. So what we did is we collected a panel of cell lines, and now in 2020, this panel looks pretty small, but at the time, it was very difficult to genotype tumors for these type of mutations. And we were able to identify a group that had BRAF mutations in the tumor cells, others that were RAS mutated, which is the protein just upstream of BRAF that activates it, and then a set of tumors that had activation of this BRAF MAP kinase pathway, but due to further upstream alterations, things like HER2 amplification or EGFR alterations. And then we just compared these three groups in terms of their dependence upon the pathway. And we didn't have a good BRAF inhibitor at the time, but what we did have was a very selective MEK inhibitor, which is the proximal downstream target of BRAF. And what was quickly apparent was that tumor cells that had a BRAF mutation were addicted to this pathway. They were very sensitive to MEK inhibition. Tumor cells that had RAS mutations were variably sensitive, and that's in part because as you go up the pathway, while you may be activating downstream, there's other pathways that are also coming off these oncoproteins that may be diminishing addiction upon the downstream arm of a pathway, like the RAF, MEK, ERK downstream effector of RAS, or RTKs can activate RAS as well as PI3 kinase and RAF and RAL-GEFs and other downstream pathways. So tumors that are RTK dependent are usually not dependent upon MEK alone. So this type of pattern where some subset of tumor cells with a specific mutation were dependent upon the mutation, whereas most others were not, is really what we're looking for with precision oncology. Because if you have a drug that works in every cell, every cancer cell, it's probably going to kill a lot of normal cells or harm a lot of normal cells, and it's probably not going to have selectivity of the tumor over the normal. So because this is somewhat old, just to make a long story short, eventually we developed good inhibitors of RAF to go along with our good inhibitors of MEK. And we started to take these into the clinic and see these type of dramatic responses. And so this was a set of trials led by Keith Flaherty and Paul Chapman, Keith now at Mass General, Paul here at Sloan Kettering, showing these dramatic responses in melanoma patients here by PET scan, here by CAT scan. And then that led to improvements in progression-free survival, which I'm showing here, as well as overall survival. But as with almost all the precision oncology approaches that we see and have developed, we see variable response among patients. So some patients respond very well. These patients here are getting complete responses to the treatment. Other patients didn't respond at all. And then some patients in the middle had partial responses. And so what a lot of my lab has done over the last few years, which I'm not going to have much time to talk about today, is what are the co-altering factors with BRAF, for example, that is diminishing addiction to BRAF, that is conditioning the response to these drugs, and can we use that knowledge to now develop rational combination strategies. But I want to talk about single drivers mostly today, and another way that we've really tried to identify these single mutation targets that we could use drugs selectively for individual patients is to use what's called a phenotype to genotype approach. And what that means is that we try to take patients who have responded to drugs, sometimes dramatically and sometimes in a unique way, try to understand what it is about their set of mutations that's leading to that particular phenotype. And we call these patients, these outlier patients, if they're the rare responder in a sort of a much larger population of patients who don't respond, extraordinary or exceptional responders. And really the paradigm case we had for this was a patient with bladder cancer who was on a clinical trial of everolimus that was being run by Matt Malowski at Sloan Kettering about a decade ago. He's now down at UNC. And so this was a trial that was based upon not the strongest biologic rationale, but sometimes when you don't have a lot of strong biologic rationale drugs, you try things that might not sort of seem to be as promising. And so with everolimus, this had been a drug that was approved in kidney cancer, and so maybe it would work in bladder cancer as well. And so we designed a clinical trial to test 40 or so patients, giving them everolimus, which is a pill. You take it once a day. And the bar was pretty low back then. There were no FDA-approved treatments for bladder cancer. And what we were asking for was, could we at least delay progression for those cases? And we enrolled 45 patients, and unfortunately only 2 of them met the endpoint that we were looking for. Only 2 of them responded well enough over 6 months that we were excited about that. And so given that result, this was a statistically negative study, and the thinking was, let's move on to the next, hopefully better idea. But one of these 2 patients really drew our attention. She was in her 70s, and she presented in 2009 with metastatic bladder cancer to lymph nodes. And so we went ahead and we do what we do in those patients. We gave her chemotherapy. We were able to shrink down her cancer partially, and then took her to surgery. And she was briefly cancer-free after the surgery, but unfortunately, her cancer then came back. She then had this lymph node involvement, which back in 2010 was pretty grim news, because we didn't have any additional therapies that we could offer such a patient that had been approved by the FDA. And so we really encourage these patients to go on clinical trials. And even though this clinical trial, wasn't based upon the strongest biology, and even though not almost any other patients on the trial responded, this patient responded dramatically. So here's her scan in 2010. Within 3 months, her scan had dramatically improved. And within 6 months, it had resolved, the cancer had resolved completely. And then a year later, she was still in remission. And actually, 9-10 years later now, she remains a complete responder to this treatment. So this type of outlier response is seen commonly on many of our negative studies. But historically, we didn't have the ability to figure out, was there a genetic mutation that was the real basis for this addiction to the target of everolimus. And so back in 2010, we did the type of assays that we had available. We did a hotspot assay called Sequenome, looking for hotspot mutations like the BRAF one I mentioned to you, and didn't find any mutations. We did what's called a RACE-CGH, looking for copy number alterations across the genome, and discovered no mutations. But fortunately, there was a real jump and leap in the technology, which allowed us to perform whole genome sequencing of this patient's tumor and normal DNA. And in fact, this was the first whole genome sequence ever done at Sloan Kettering, and the first one ever done worldwide in a patient with bladder cancer. And it was notable for the fact that the patient had a lot of mutations. There were actually 17,000 somatic mutations that were in the tumor that were different than the patient's normal DNA sequence. And you ask, if you have that many mutations, how could you ever know which of those were important? But it's a real testament to how much we had learned about the human genome from the Human Genome Atlas, how much we had learned about different genetic mutations in tumor cells for many retrospective studies, that we were able to very rapidly map all 17,000 of these mutations to the coding sequence of the patient's DNA, ask which of these mutations were located in the coding regions of genes, which ones were not, And if you just do that simple analysis, only 140 mutations were located within actual coding sequences of genes. And then if you look at each of these 140, what we call non-synonymous coding mutations, there were really two that jumped out of us. One, a frameshift mutation in TSC1, the other nonsense mutation in NF2. And the reason that these were interesting was that loss of function of both of these genes activates a protein complex called TORC1, which is a set of enzymes, which is the direct target of everolimus. So in retrospect, this patient was the perfect patient for everolimus because she had a set of mutations that led to her tumor activating the target of the drug. The difficulty is that we just didn't know that when we put her on the study. We put her on the study having no idea what her genetic makeup was. And we didn't even know at the time, back in 2012, when we published this, that TSC1 was even mutated in patients with bladder cancer. There hadn't been a TCGA effort in that disease yet. But one could imagine that if we knew that information prospectively in this patient and in other patients with bladder cancer, maybe we would have done this trial differently. Maybe we would have specifically enrolled her onto the study because of her genetic mutations, and maybe we would have put many of those non-responding patients onto other clinical trials. And so this is the paradigm for these extraordinary responders. We take these outlier patients, we take tumors from them, we do some sort of molecular profiling to come up with a hypothesis as to why those patients maybe responded. We always go back to the lab to study that, to see if that makes sense. But what we needed is we needed a test that would allow us to prospectively, in the clinic, identify these mutations so that we could inform the doctor of the mutation and inform the patient of the mutation and use that information to guide that patient's care, either their care into a standard of care therapy or their enrollment onto a potentially promising clinical trial that is promising in that patient because of their unique tumor genetic makeup. And so to be able to do this, we had to write a clinical trial because we needed consent to be able to do research sequencing, to be able to report research results back to patients, as well as we needed consent to do this testing for free because when we started this program back in 2013, 2014, genotyping was not something we could bill as a standard of care diagnostic test. It hadn't been proven, at least in most cancers, to help improve outcomes for patients. There were some tumors where that was the case. Lung cancer, it was standard of care to look for EGFR mutations, later ALK fusions, and now 7 or 8 other alterations. In melanoma, it was standard of care to look for KRAS, and in melanoma, BRAF. But these were the minority of tumor types, whereas in most cancers, prostate, breast, all GYN cancers, other GU cancers, leukemia, these type of drug targets had yet to be validated. So what we did is we wrote a clinical trial, and I wrote this with Dave Hyman and Mark Robson, and Sophia Stadler has now come on in later to take a big role into this, where the goal was very simple. We wanted to, for every single patient who came into our clinic for care, we wanted to be able to figure out what were the mutations that were driving that patient's tumor in terms of its formation, in terms of its growth, in terms of its resistance to standard of care treatment. And we wanted to be able to do that in a very efficient way, return the results back quickly, and then use that information to guide their care. And I'll tell you in a second how we do this, but we're able to, at the same time, identify germline mutations. These are mutations in the patient's normal cells that may have led to their cancer development, to begin with. And we needed to have the patients consented on this protocol to return those results as well. And then in patients who opted in to this effort, we also consented them to do other research analyses, broader than the targeted panels we use in the clinic, things like whole genome sequencing or RNA sequencing in terms of whole transcriptome RNA analysis, and now newer technologies like single cell sequencing. So again, to start this, we didn't start with the assay, we started with a clinical trial that would allow us to answer questions that would eventually, hopefully, show that this technology was helping patients and thus should be used in a broader number, or essentially in all patients, hopefully in the future. So we then needed an assay, and this here we were fortunate in that Mike Berger had recently been recruited to MSKCC by the pathology group, and we established a relationship with diagnostic molecular pathology here at Memorial to take that assay and to validate it for clinical use and then to translate it into use routinely, prospectively for patients. And so briefly, how we do these assays, these are called targeted capture assays, because instead of sequencing the entire genome, which would be very expensive and would take very long to do the analysis, we sequence just a small part of the genome. And so what we do is we take the tumor from the patient, usually just an FFPE slide that was processed at the time of their surgery or at the time of their biopsy. We don't have to do a new biopsy to do this type of analysis for most patients. And then we take their blood, which is a source of normal DNA. And to be able to analyze just a subset of the genome, we develop these baits, we call them, which have a probe on them, which hybridize to the parts of the genome that we are interested in, the parts of the genome that are the cancer-associated genes where these type of mutations are found. And then we use beads to pull down that part of the genome, wash the rest of the genome away, and then use a next-generation sequencing machine to analyze that tumor genome as well as that patient's normal genome, so we can compare the sequence in the tumor versus the normal to find areas where there's a mutation, a change in the DNA sequence that's in the tumor cells but not in the patient's normal cells. I think one of the great things about our program is that we try to make this data available broadly in a anonymized way to our researchers here at Sloan Kettering, as well as researchers throughout the world through a program called ACRgeni. And the way we facilitate that de-identified, de-anonymized data sharing is through a program called CBio Portal for Cancer Genomics. And I made this slide overnight highlighting how large this program has now grown over the past 6 years, in that we've now sequenced over 55,000 tumor samples from over 46,000 patients. And so you can see here there's more tumors than patients, because increasingly we have patients where we've sequenced their tumors from different parts of their disease course, maybe a tumor at the time of diagnosis, and then maybe a tumor a few years later at the time of their recurrence. And then there's some patients who have actually had multiple cancers that we can actually compare the genetic sequence of going forward. This is the view in the CBio portal that shows the patient, a specific patient. This was a 30-year-old patient with a brain tumor who sadly died of their disease. And this genetic sequencing identified a set of mutations, in particular this IDH1 mutation, for which there's now an FDA-approved drug for certain cancers available. And we've started to annotate the known knowledge about these mutations using a program and a website. It's all available on a website called Unko KB. And so we have a scaling system for these mutations, telling us that what is the clinical evidence that that mutation is a predictive biomarker for a particular drug. And this little flame here shows that it's a hotspot mutation, one of these mutations that recurrently arise in multiple different patients. And then the website even has descriptions about the biology around these mutations. How does the mutation lead to tumor formation, and what drugs might be available for that particular cancer mutation. We've also added additional value to this research data set. So for example, the patient's H&E slide, which allow people to develop potentially machine learning algorithms that would identify factors within this the morphology of these tumors that might, together with the genomic data, tell us better how to treat that individual patient. So I just wanted to give two quick anecdotes of how we've used this clinical genetic data set to identify new biology and help us understand why some patients respond and other patients don't. And so this was a collaboration which was led by Padron Razavi, who is a junior clinician scientist here at Sloan Kettering, together with Jose Baselga and Barry Taylor and many other investigators, where we wanted to ask the question, why do patients become resistant to hormonal therapy who have breast cancer? And so that's one of the commonly used treatments for patients with metastatic breast cancer, cancer that's spread and recurred, is that you treat them with these hormonal treatments. And oftentimes those patients respond dramatically for many years, but eventually almost all of them develop resistance. And so why? And so what we did is we took this genomic data set, and then Padron very carefully reviewed all of the medical records of these patients and asked the question, were the tumors collected before the patient received any treatment, or was the tumor collected after they had progressed on hormonal therapy? And then asked the question, are there some mutations in the progression samples that are not present in the pretreatment samples? And so the untreated samples are in gray. The progression samples are here in black. And you can see there's this gene ESR1, where about 20% of the tumors that were collected at the time of treatment resistance to hormones have an ESR1 mutation, where that's almost never found in the pretreatment tumor samples. And ESR1 encodes for the estrogen receptor, which is the direct target of many of these hormonal therapies. So this was actually known prior to this study, which was reassuring that we found what we were supposed to find. But what we also found is that there was this whole set of other mutations that were less common than ESR1 mutations in the progression samples, but were mutually exclusive with ESR1 mutations. So you either had an ESR1 mutation in the tumor or one of these other many mechanisms of treatment resistance, which were all mutually exclusive with each other. So again, this is a use of this type of prospective clinical data set to identify a mechanism of why patients will fail therapy. And now that we know that information, we can now use it to develop drugs against these other mutations that might prevent or delay the emergence of these drug-resistant tumor cells. Another example is a collaboration we did with Jim Harding and the GI group here at the Medical Oncology group at Sloan Kettering. And in the tumor that Dr. Harding is very interested in, hepatocellular carcinoma, there was encouraging early data with immune checkpoint inhibitors, but unfortunately only a small number of patients were responding. And so what we ultimately saw by integrating the genomic and the clinical data from this data set was that if you had mutations in this Wnt beta-catenin pathway, you never responded to immune checkpoint inhibitors. And so this is a waterfall plot showing the degree of response. All the responders were in the Wnt beta-catenin wild type group. It doesn't explain all of the resistance, because here's some patients who didn't respond that were wild type, but the patients who had these that were wild type for these set of mutations had a much longer progression-free survival on immune therapy versus those that didn't. So this is an example where we're not finding the mutation that is sensitizing to the treatment. We're finding a mutation that causes resistance. Here's an example where we're actually using the data prospectively to guide a patient's care. This was a patient of Alex Shashari's with melanoma who had been treated with immunotherapy but had become resistant to those therapies, had tumor genomic sequencing, and what we noted was that this patient had very high level amplification of HER2, the HER2 gene, which encodes for HER2, which is a known drug target in breast cancer, endometrial cancer, esophagogastric cancer. And this type of high level amplification correlates with response to certain treatments that we have in those diseases. And so here's that patient's scans. They had a tumor here in their lung, which had caused a lot of fluid to accumulate here and a collapse of that lung. The patient was then treated with a drug called TDM1 Herceptin, which binds to and delivers a chemotherapy drug to the HER2 molecule that this gene encodes. And you can see just within a few weeks, that tumor had shrunk down and the patient had a complete response. And we call this what's called off-label use of a drug. It's when we use a drug for an indication where the drug's approved for something else, but we're using it for some biologic rationale for a different cancer type. Now, the problem with this is that these drugs are oftentimes very expensive and the patients can't get access to the drugs. And so we've tried to develop a series of clinical trials that at least solves in part that drug access question by making a drug available to a patient with a particular mutation, no matter what cancer type they had. And we call these basket studies. And this was a study I was fortunate to lead with Dave Hyman, where we took patients with HER2 mutations and then treated them with inhibitor of HER2 called neratinib. And these studies, we have different baskets, depending upon what type of cancer they have, a basket for bladder patients, colon cancer patients, endometrial cancer patients, et cetera. And we always have an other basket here for patients with other tumor types that we've never seen this mutation, but we want to allow those patients to have access as well. And we sometimes see very dramatic responses to this treatment. This is a woman with breast cancer who had a HER2 mutation in the kinase domain, who had many areas of metastatic disease. This patient had received 8 lines of prior systemic therapy, the last 4 of which she didn't respond to. She was then treated with neratinib and went on within 2 months to have this kind of complete response. And so the dilemma with this paradigm, again, is that it's not standard of care necessarily to test for HER2 mutations. So we needed to have a clinical trial to allow those patients to be tested as a research test back in 2004, 2014, 2015. And then we also need to have a clinical trial that patient could go on that would provide access to that drug in an investigational setting. So what have we learned from these basket studies? We've learned that in some cases, the drugs can work in a broad range of tumor types, but in most cancer types, cancer type matters, not just mutation type. So what we find is that the likelihood of responding to neratinib, if you have a HER2 mutation, varies as a function of what kind of cancer you have. You're much more likely to respond if you have breast cancer than, for example, if you have colon cancer. And we haven't figured this out completely, but in part it's due to different co-mutations in different cancer types, different lineage-specific developmental pathways that diminish addiction to a particular target. But it's an active area of investigation within my lab and within many other labs throughout the world. But one of our major challenges is which HER2 mutations should we put on the trial? Just those that are known to be activating, those that are hotspots. Can we use computational algorithms to select which mutations go on? And that's really a major focus of what my lab has been trying to do over the past 4 or 5 years. And these are just some of the ways that we try to do we try to use more of these extraordinary responders. We try to use big cohorts like TCGA, try to use computational approaches and what we call co-clinical trials or lab-based screens, and just go through each of these individually. So here's one of these extraordinary responders. This was a patient with ovarian cancer who had a tumor here in their abdomen. This patient had failed the standard of care treatments and then went on a clinical trial of a drug called AZD6244 and went on to have a good response, which has been durable now for over 10 years. And the question is, why did this patient respond? We went ahead and we sequenced their tumor, and we found that this patient had this deletion here in a gene called MAP2K1. And the reason that deletion was very interesting to us is that the gene that this patient received, called AZD6244, was a direct target of this MAP2K1 gene, which encodes for a protein called MEK1. And so MEK1, here's the crystal structure of it. Here's where this deletion is located. And so it's in the part of the protein that's called the negative regulatory helix. It folds over onto the protein. And when you mutate this part of the gene, you open up that protein into an active conformation. And we always go back and we validate that kind of hypothesis in the lab. And so here's that deletion mutation. We went ahead and we made it. And you can see here, it leads to activation of ERK by this Western blot. And so that's one way we can identify new mutations that have never been seen before by linking them up to a phenotype from a patient who responded. Another way is looking at recurrence. This was a hotspot algorithm developed by my colleague Barry Taylor, looking for hotspots that were specific in particular tumors or pan-cancer hotspots. And to me, from my experience, the thing that's most predictive of something being biologically important is its recurrence or hotspotness, you could say about that. We've also just done this by brute force. This was an effort led by Afrothini Hanrahan, a junior scientist in my lab who's now a faculty member. She was junior when she started, she's a faculty member now. It's been going on for a while. Whose goal was to try to make every single MEK1 mutation we found in every single patient we sequenced using this perspective sequencing initiative. And you can see some of these are activating based upon this Western blot, whereas some of them you see no band here are inert mutations. And if you have one of these MEK1 mutations, really what you need to know is my mutation important? Is it activating? Is it sensitive to a drug? So you can make all these mutations, which takes a huge amount of time and would probably not be feasible for every mutation that we identify, because we identify thousands of mutations in many tumors that are hypermutated. Now we're trying to use an approach called 3D dynamic simulation, where we can look at how a known activating mutation or phosphorylation changes the conformation of the kinase in 3 dimensions, and then model that with other mutations to show that they either activate the kinase, they're predicted to activate the kinase, or predicted to be inert. And we see a very strong correlation in some genes between these predictions and what we see when we go to the lab and actually make the mutation. So I want to spend now the last sort of 15 minutes of my time on really saying where we're going next, how we're trying to expand the population, make the platform better, some outreach efforts we have, how we're trying to study cases that are not solved, and talk briefly about cell-free DNA. So this was a collaboration with Ellie Van Allen and others up at Dana-Farber and the Broad, where we wanted to ask the question in patients with bladder cancer who get neoadjuvant chemotherapy, so chemotherapy before surgery, why do some of those patients respond whereas others are resistant to the treatment? And so what we did is we took the outliers. We took the patients who had a complete response. We took the patients who had no response, and we did whole exome sequencing of those outliers, and then asked, are there genes mutated in the responders but not in the non-responders? And when you do that analysis, there's only a single gene that arises from that analysis called ERCC2. And in retrospect, that made a lot of sense because mutations in ERCC2 sensitize you to chemotherapy, especially cisplatin, which is the type of chemotherapy that these patients receive. That was known biology. What we didn't know at the time of this study was that ERCC2 was mutated in bladder cancer and that it was potentially the reason some patients responded so well. So based upon that, we've developed a clinical trial where patients with newly diagnosed locally advanced bladder cancer will receive, they get local surgery to cut out part of the tumor through a cystoscopy, but not the entire bladder. They then receive chemotherapy to shrink down or completely eliminate the tumor. And then in those patients who get a good response to that chemotherapy and have no additional tumor left in their bladder based upon staging studies, who have these mutations, we're going to offer them bladder sparing. And this is based upon some early results we have that sometimes patients will get the chemo and then have these mutations, then they're not well enough to get the surgery, but then they still seem cured years later. So we're hoping to use this genetic data, not in patients with advanced metastatic disease, but in patients at the time of diagnosis to try to minimize the negative consequences of their treatment, try to minimize the amount of surgery or potentially chemo or radiation that they need going forward. Of course, this is challenging because not every ERCC2 mutation is the same. They usually cluster in this particular domain, and we don't yet have enough sequencing data yet, even though we have 50,000 plus patients, to know where the key hotspot mutations are. So we've actually worked with a set of investigators led by Kent Mao and others at Dana-Farber to develop a high throughput assay, which allows us to test each of these mutations, maybe not prospectively, but retrospectively, to ask whether they impair this DNA repair function of ERCC2, which you can see in these mutations. These are mutations that are similar to the wild type protein in that they're inert. They don't reduce the DNA repair capability of the cells that they're found in. We're also trying to make the platform better by adding more genes that have been discovered since its initial development. But what I'm most excited is we're going to add DNA probes against viral DNA sequencing. And that was based upon one of these extraordinary responders that was analyzed with Yelena Janjigian, where we had a patient with gastric cancer who has had now a complete durable response over 4 years, maybe 5 at this point, with gastric cancer to immune checkpoint blockade, even though the vast majority of patients with this disease do not benefit from that therapy. And what it turned out was that this was the only patient in the cohort who had an EBV driven tumor. So EBV is a virus that causes a flu-like syndrome in usually teenagers when they get it. But what happens is sometimes the EBV integrates into the human genome and causes a set of mutations that lead to cancer. But also EBV integrated tumor cells can express some of the EBV proteins on the cell surface, and the immune system can recognize those viral proteins and help clear the cancer cells. And immune checkpoint blockade seems to work particularly well in some of these virally driven proteins. So by putting probes against the viral DNA into our tumor sequencing assay, we can screen all of our patients for these virally integrated proteins, and then potentially do a basket study where maybe every patient who has an EBV tumor would be eligible for a particular immune type therapy going forward. We're also trying to get more data out of the information that we've already generated. As I mentioned, we can call germline mutations. These are mutations that people are born with that might have contributed to their cancer formation. And given the lack of time I have, I just want to focus on one gene called BRCA2. This is a gene that was initially discovered that when mutated in certain families was associated with early onset ovarian and breast cancer. But what's notable about our cohort, and this was an analysis of over 8,000 patients here so far, was that you were more likely to have a BRCA2 mutation in your germline, in your normal cells, if you had pancreas or prostate cancer than if you had ovarian and breast cancer. So it was routine to test for these germline mutations in ovarian and breast cancer patients, but not in pancreas and prostate. And based upon this data and a set of publications that have followed this work, we now routinely believe every patient with pancreas and prostate cancer should be tested for the BRCA2 and other germline mutations that may explain why that individual got their cancer, and also give them other treatment options going forward. We're also trying to understand whether the mutations that we identified are found in all the cancer cells or in just a subset of cancer cells that were analyzed. And we call that concept clonality. So if a gene mutation is clonal, it's found in all the cells in the tumor. If it's subclonal, the tumor's heterogeneous. The mutation might be found in some parts of the tumor or some tumors and not in other tumors within that patient. And what we noted on the neratinib study was that all the patients who benefited from the drug had clonal mutations, which makes sense. If you've got a tumor that's mixed, where the target that you're going after is in some cancer cells but not in others, you might give the drug and then very quickly select for the tumor cells that don't have that mutated target that's sensitizing. But it's more complicated. We still don't yet know how to use this information. And just here's an example of a patient who did benefit clinically who had a subclonal mutation. So this was a patient who was on a trial of an AKT inhibitor who had an AKT mutation. And we were fortunate to have multiple tumors that had been collected from this patient. And we found that the clonality varied as a function of the different tumor sites. So this was work led by Dave Hyman and in collaboration with Barry Taylor. And what was notable was that the patient had a mixed response. Some of their tumors responded, others didn't respond as well. But the tumor that responded the best was the one with the highest clonality. So at this point, we can't say that subclonal tumors are not going to respond, but this is clearly something that's influencing why some patients, even who have the same mutation, are responding differently to other patients with that same exact mutation. We're also trying to integrate what we call mutational signatures, which is the type of mutations that are found within the cancer, not just looking at a single mutation, but the type of DNA changes that are found. So for example, different toxins or different inborn mutations can lead to different mutational signatures because of how they affect DNA repair or how the toxin damages DNA. So this was a patient of mine with prostate cancer who had this MSK impact report, and it found a lot of mutations. And that was what was notable in prostate cancer because we don't find a lot of mutations. And based upon that high mutation rate, he ultimately went on a clinical trial of an immunotherapy combination, which had an anti-PD-L1 therapy led by Matt Hellman, and went on to have a complete response. That was almost 3.5 years ago. And he may even be cured of his prostate cancer. And this was a prostate cancer that had spread to distant sites, that had failed the standard of care treatments, and would have likely been fatal within a pretty short period of time. And this is a gentleman who's now years out because the genomic sequencing identified a mutational signature which suggested a treatment, in this case immunotherapy, to which most patients with his disease wouldn't respond, but he ultimately did. So we've now taken a look at this phenotype in great detail in patients with prostate cancer, and we find it in only 3%. So you have to sequence 100 patients to find 3 who have this sequence. And then when we give the immunotherapy, we only see about half of them respond. So it's finding a little bit of a needle in a haystack, but if you are the patient with an MSI high tumor and your tumor is not profiled, you may miss out on a potentially curative or effective treatment for your disease. And really, some of the debate in the community is, should we, is this cost effective? Should we sequence lots and lots of patients to find these things are rare? And so, my argument in this case is absolutely for this scenario. But for the other scenarios, I think it's beholden to us, the people who are pushing this and developing this technology, to try to make this technology cheaper so we can make it available to more patients. I'm just going to skip over a couple things due to time. I'd also recognize that not everyone has access to this technology. And what we've seen is that that disparities in genetic testing may increasingly lead to disparities in clinical outcomes, which is something that is a great area of focus for our hospital and for many others. And so we've worked with Carol Brown here at MSKCC, one of our surgeons, to create a program called Impacted, which is Impact to End Racial Disparities, where we offer free genetic testing to patients who come to Queens and Kings County hospitals. These are city-supported public hospitals where the vast majority of patients being seen lack health insurance and oftentimes don't have access to these technologies. And so we need to bring these newer technologies to those patients and make sure there's universal access to these type of profiling efforts and drugs that are selected based upon those results. We've also worked with patient advocates, in particular my wife, for some of this, to create a program called Make an Impact. And so what this tries to do is try to understand better the biology and genetics of rare tumors, maybe tumors that we don't see enough at Sloan Kettering to be able to figure out alone, that we need to work with a much broader community. Maybe these are tumor types where there's 5 or 10 or 15 patients in the entire United States alone who have these cancer types. And so it just wouldn't be feasible to wait for enough of those patients to come to us. Well, it's 2020. We've got social media. We're more connected. Why not reach out to those patients where they are, the institutions that they're located in, and ask them to send their cancers to us so that we can offer them this genomic profiling for free. So been able to do this for every tumor type, but there's several tumor types you can see listed here where we've done it. For example, these ovarian germ cell tumors, where there may only be about 100 cases in the US a year. A place like Sloan Kettering might see 2 or 3 of these a year that require chemotherapy. And so we really need to reach out to those patients. And why not use social media, again, to bring those tumors in without requiring the patients to come see us. Another place where we've done this is these mammary secretory carcinomas of the breast. The reason this one was interesting was that we had a drug that inhibited a gene that was usually mutated in these cases called NTRK. And we had an example here. This was a young girl from Bangladesh who at 8 years old developed a tumor much smaller than the one I'm showing here on her chest. And they cut it out. It then recurred 2 years later when she was 10. They cut it out again, and then it came back again. And you can see she had this very large unresectable tumor on her chest. And we heard about this patient through our social media outreach efforts. We're able to get the tumor to send here to New York. We were able to sequence it, identify this mutation, this TRK fusion in her tumor, and then working with our pediatric oncologists, in particular Neil Shukla and Dave Hyman, and Alex Drillin, who was running the clinical trial of this drug. We were able to get this patient this drug, which is a drug that you take just once a day. And you can see within 3 weeks, she had this near complete response. She actually remains on this drug to this day back in Bangladesh, where she is. She had to come briefly to New York to start the treatment, but we've been now able to manage her back in her home country. So this is a case where, again, we can make the world smaller by using the tools we now have, the internet, the communication advances that we've had to essentially try to offer these treatments to patients anywhere in the world who may have a particular mutant type. We're also cognizant that a targeted approach such as MSK Impact that looks at a little less than 500 genes is not going to solve every case. This is an example of that. Here's a patient with melanoma who got MSK Impact. We found 11 mutations. This TERT promoter one is an important functional mutation, but not enough to get a melanoma. And these others we think are just benign variants. And so we try to study these cases. We call them cancers of unknown driver in greater detail. So what we did is, under this research protocol, we did whole exome sequencing. We now found about 40-fold more mutations in this case. And unfortunately, that wasn't informative. So we analyzed the patient's RNA using what we call this Archer fusionplex assay. It can identify fusion genes in several dozen cancer-associated genes that are oftentimes involved with these activating fusions. And this patient had an NTRK fusion that wasn't detected by DNA sequencing, but was detected by RNA analysis here. And fortunate for this patient, we actually had a clinical trial available for them. And here's their PET scan. This patient had a melanoma that started here in the leg. These are all melanomas that had developed along the patient's leg. And within 2 months of starting this drug, initially called LOXO-101, this is now an FDA-approved drug called larotrectinib, this patient went on to have a dramatic response, as you can see by the CT scan. Another example of this unknown driver effort is a set of studies led by Omar Abdel Wahab, who generously introduced me in patients with histiocytosis, where we had already discovered that BRAF mutations were common, MEK1 mutations were common, RAS mutations were less common. But he identified that there was a subgroup that had mutations in a gene called CSF1R that was mutually exclusive with these other known oncogenic events, and went on to show that these are activating mutations. And there's in fact a drug that potentially could work for this subset of cases that he's now working on. But there's still about a group of 10% of the patients with this rare disease where we still haven't solved them. And we still need to analyze those tumors in greater detail to understand better for every patient, even if it's rare, why did that patient get their cancer? What's making it grow so that we can develop rational therapies against those particular genetic mutations. The other dilemma we have is that we can't get the DNA in some patients. And there's now been this incredible advance in using what we call cell-free DNA to detect very tiny amounts of the tumor DNA, which is shed from the tumor cells into the blood, into the plasma. We can then draw the blood from those patients, sequence those DNAs using very, very sensitive technology, and identify these potentially actionable alterations. And the case that really jumps out at me is this case. This was a patient, very young gentleman in his 20s, that had an unknown primary cancer. And given his age demographic, it was an unknown primary. You couldn't tell what it was under the microscope. They thought maybe he had a germ cell tumor. So he was referred here to Darren Feldman for treatment. And we tried to run MSK impact on that patient, but it failed. We drew his cell-free, really doing a research effort validation at that point. About 2 months later, we started to get some results on that, but it was still incomplete. He then progressed on the chemo we give for germ cell tumor. We then were able to biopsy his tumor more easily in an adenocarcinoma. So we never really had a germ cell tumor. We weren't able to make the correct diagnosis in him given the limited material that we had. And then unfortunately he died a few weeks later, only 3 months after his diagnosis. And then shortly after he died, using the cell-free technology, we were able to figure out that he had an EML4 ALK fusion. So this is an example of a patient who had a fusion that we have a drug for, but we weren't able to, in the time frame that we had, figure that out, in part because we couldn't get an adequate biopsy that had good enough DNA from the tumor from this patient. And that happens because sometimes the tumors are very deep within the body, and we put in needles and we still can't pull out enough tumor DNA to do these analysis. And cell-free DNA gives us the possibility that maybe in a non-invasive way, just by drawing the blood, we can detect these mutations. So how do we do that? Well, the real limitation of using cell-free DNA is that there's a lot of noise when we do the sequencing. There's noise that's created when we do PCR to generate the sequencing library. Sometimes the DNA has been damaged by oxidation as we're doing the processing. And then as we do the sequencing, we sometimes introduce errors into the DNA sequencing. So to be able to do this cell-free analysis, we have to reduce the errors, and that's what we do. We now have developed a method to really strip out most of the errors that are generated through this process. And there are still some errors. They're just about 100-fold less than we see with the traditional tumor sequencing methodology. And the way we do this is complicated. I don't have time to explain it. I'm happy to explain it by email or by message later. But we barcode the DNA first, and that allows us to sequence many, many copies. And that's why it's more expensive to do cell-free DNA sequencing per gene, and then use a consensus sequence to identify what are the real mutations versus what are the errors in our sequencing output. And we've now developed an assay that's in clinical use, was translated into diagnostic molecular pathology. We've now used this assay in over 1,000 samples so far to date. And we're hoping to use this assay to match patients up with treatments, to better understand heterogeneity, and also to do a set of trials that use MRD, minimal residual disease, to treat patients earlier before they recur. So just to mention two of those, and that's the end of my talk here. This was a very interesting patient where the cell-free suggested heterogeneity that we wouldn't have otherwise known. So this was a patient who had their primary tumor biopsied and had a set of mutations identified by tumor sequencing. We then biopsied their metastasis, the tumor that had spread to the patient's distant sites. And we found most of the same mutations in the MET versus the primary tumor, but we found a druggable FGFR3 mutation in the metastasis that we could then use that information to give the patient a drug called erdafitinib, which is approved for patients with FGFR3 mutant bladder cancer. The day before we started the treatment, we drew this blood sample for cell-free DNA analysis, and we were able to detect that FGFR3 mutation, but also other FGFR3 mutations in the plasma. And what it appeared to be is that those FGFR3 mutations were all subclonal. So what was happening here is that this is a late onset FGFR3 mutation that's not in the primary tumor, it's in the metastasis. And cell-free allows us to do that in a way that's very difficult. Otherwise, we'd have to biopsy multiple sites, which is not feasible. By drawing cell-free, you can get a sense for maybe all of the different cancer sites in an individual patient. The other thing we're going to do with cell-free is we're going to use it to do these MRD studies where we take patients who get their standard curative intent treatment, We then follow them for evidence of microscopic recurrence of their disease using cell-free DNA, and then hopefully can start using the cell-free DNA positive information. If we see their tumor mutations in their blood, we can start them on treatment earlier than we would otherwise. Instead of waiting until the tumor was bulky and was visible on x-rays, maybe we can start it earlier when it's microscopic and actually cure those patients versus just give them treatments that are going to delay their ultimate demise from their cancer. And so this is called adjuvant therapy, where you give something to a patient where you can't visibly see any evidence of recurrence. We've shown this works for breast cancer and for many other cancer types, but cell-free may allow us to know which of the patients who need additional systemic therapy, maybe avoid this toxic chemotherapy in some patients who don't need it, and give targeted adjuvant therapy in patients who don't. So this is my concluding slide. I just want to say that precision medicine is often criticized because not every patient has a targetable mutation. I would comment that the next-gen sequencing assays we do are not a therapeutic modality. They're just a test that allows us to identify the presence or absence of these druggable alterations. It's a test that's now NGS replacing a lot of older tests. In many cases, it's more sensitive. It gives you more information. It can allow you to test multiple genes at the same time, which ultimately could make the testing much more inexpensive. But even today, we only identify really actionable alterations that have clinical evidence of actionability in about 40% of our patients. And in that other 60%, in most of them, we do identify oncogenic alterations. We just don't have drugs for them. So for mutations like KRAS and RB1 and NF1, we know these are the drivers of many tumors that we see in our patients, but we don't have therapies that can inhibit tumors that have these mutations selectively. And broader testing is probably not going to help for these patients. It helps for that very tiny number where we find no druggable oncogenic mutations. But for most patients, it's not going to be broader sequencing that's going to make dramatic progress over the next few years. It's going to be better drugs. And so essentially, I'm joining with many of my colleagues going forward, hoping that we can see many of these drugs come out over the next few years. So I just want to thank the many people who are involved in this effort. I'm just highlighting a few of the leaders of this program, but many of these anecdotes had teams of dozens who were involved in this sort of clinical translational type of studies. And I'll hand it back over to Omar now to see if there's any questions, and you can also ask your questions later on that are emailed to us. So thank you again for the opportunity, Omar.
Yeah, no, thanks, Dave. It was a great talk. Can you hear me okay?
Yep.
Okay, great. Well, there's a lot of excitement because we have about 20 or so questions. We don't have enough time to get through them all. I'll ask you just a couple, maybe the ones that I'm most interested in. Maybe there's actually a couple from patients as well. We'll try to get at least one of those.
And we'd love to have those patients, reach out to me other ways. We could answer those in other, ways.
Yeah, so I know Dave is on Twitter, for example, so if anyone can find him there. All right, Dave, first question for you. This one I want to ask you as well. Curious what you're going to say. How often are you able to provide treatment that you otherwise wouldn't based on MSK impact testing? Like how often does it actually impact the treatment we're offering, you think?
Yeah, there's some cancers like lung cancer where almost half of the patients now, if you have lung cancer, there's a specific targeted therapy that you match up for that particular mutation. And that the first of those was EGFR mutations back in the early 2000s. Early to mid 2000s, where that was discovered as sensitizing to EGFR inhibitors, but that was followed by ALK fusions and ROS fusions and BRAF mutations and more recently RET fusions and several others now that are also sometimes in clinical trial like KRAS. So in a cancer like lung cancer, it's closing in on the majority of patients. That would be true as well with tumors like GIST sarcomas. But for other cancer types, it's the minority. For prostate cancer, for example, there's really two main things that we look for. We need to look for microsatellite instability, and we need to look for these BRCA DNA repair type alterations that sensitize to PARP inhibitors. So for each cancer, it's different. We've sometimes show some slides, showing a bar graph showing the ones where it's 70, 80, 90% like GIST, to others where it's 50% like melanoma, to maybe mesothelioma where it might only be 1 or 2%. And the real debate in the field is, is it worth looking for something that's only found 1 or 2% of the time? I would say the way I was trained in medical school is that, if you need, that yes, you need to find, if that's what's key for that patient, you need to find that set of mutations. The dilemma we have is that, sometimes insurers will push back and say, we're not going to pay for the sequencing. And so one of the things we've been fortunate at here at our institution is we've been able to use philanthropic donations from Cycle for Survival and the Kravis Foundation to pay for free sequencing for patients where it otherwise would not have been covered. But this is something that the field has to address. We're going to see increasing disparities of care if some patients can get this technology and others cannot.
Got it. Well, there's so many more questions. Maybe we'll ask just one more with a brief answer as much as you can, and then people just have to contact Dave because there's many that we can't.
I'm on Twitter, please reach out.
All right, so related, another interesting question. What fraction of the variants that you're finding by MSK Impact have an unknown function or unknown significance?
So the reality is most. We find a lot of mutations when we do DNA sequencing, and the number of mutations you find is directly proportional to the number of genes that you sequence. So as I showed with that one example, that cancer of unknown driver example, with exome sequencing, we find about 20 to 40-fold mutations because we're sequencing 20 to 40-fold more genes. There's 20,000 plus genes in the genome, and MSK Impact's only sequencing about 500 of those. So more genes, more variants of what we call unknown significance. Very straightforward answer. It's a variant. We don't know what it means. We don't know what it means clinically. We don't know what it means necessarily biologically. And that's one of the downsides to broader sequencing. We end up oftentimes just finding more things that we don't know what they mean, okay? And in particular for genes outside of MSK Impact, MSK Impact includes all those genes that we know a lot about. And so when we do broader sequencing, we find a lot more of these. But even in cancer genes that we've been studying for years, things like EGFR, there are still mutations that we don't know what they mean. And it's a real frustration for the doctors and for our patients. They get one of these sequencing reports back. It says EGFR mutated. Here's the amino acid change. We know there's a drug that works for some EGFR mutations. Well, what about this patient's particular EGFR mutation? So as I mentioned, that's one of the main focuses of my lab, and Omar has done a lot of this as well, really trying to figure out comprehensively using, for example, a mutational a saturation type screen, mutagenesis screen, or one of these co-clinical trials that Dr. Hanrahan and my group led, or these extraordinary responders, using the Phenotype to call this out. We're trying to pull all this data, make it very broadly available, and then we've developed a system led by Devyani Chakravarti, who's a researcher here at Sloan Kettering called Unko KB, where we try to relay this information to patients and doctors. So what Unko KB is, it tells you at the specific mutant allele level, not at the gene level, what does this mutation mean? But back to the question that was asked, for most of the mutations that we find, we still don't know what they mean.
Right. Absolutely. Well, Dave, thank you so much again for that amazing talk. And thank you all for joining us today. For our next seminar in this series, it's going to be Monday, June 8th, and we're pleased to welcome Dr. Jason Lewis. The title of his lecture will be Defying the Limitations of Antibodies for Imaging and Endoradiotherapy. Thank you all again.
Thank you.